This video is about Evergreen Revolution. The second green revolution is also called as Evergreen Revolution. Here we go and see what exactly the Evergreen Revolution is. Green Revolution refers to a series of research, development and technology transfer initiatives occurring between 1940s and late 1970s that increased agriculture production around the world, beginning most markedly in the late 1960s. The initiatives led by Norman Borlaug, the father of Green Revolution, credited with saving over a billion people from starvation, involved the development of high-yielding varieties of cereal grains, expansion of irrigation infrastructure, modernization of management techniques, distribution of hybridized seeds, synthetic fertilizers, and pesticides to farmers. The term Green Revolution was first used in 1968 by farmers for in United States Agency for International Development, that is USAID, Director William Gord, who noted the spread of new technology and said these and other developments in the field of agriculture contain the making of a new revolution. It is not a violent red revolution like that of the Soviets, nor it is a white revolution like that of the Shah of Iran. I call it as the Green Revolution. The agriculture development began in Mexico by Borlaug in 1943 based on Nazareno Stampelli's studies had been judged as a success and the Rockefeller Foundation sought to spread it to the other nations. The Office of Special Studies in Mexico became the Informal International Research Institute in 1959 and in 1963 it formally became CIMMYT, the International Maize and Wheat Improvement Center. In 1961, India was on the brink of mass famine. Borlaug was invited to India by advisor to the Indian Minister of Agriculture M.S. Swaminathan. Despite bureaucratic hurdles imposed by Indian grain monopolies, the Ford Foundation and Indian government collaborated to import wheat seeds from CIMMYT. Around 18,000 seeds were imported from CIMMYT or Mexico. Punjab was selected by the Indian government to be the first site to try the new crops because of its reliable water supply and history of agriculture success. India began its own Green Revolution program of breeding, irrigation development and financing of agrochemicals after Green Revolution. Soon after the Green Revolution or during the Green Revolution period, India adopted IR8, a semi-dwarf rice variety developed by International Rice Research Institute that could produce more grains of rice per plant when grown with certain fertilizers and irrigation. That means Green Revolution happened with the help of two independent countries. One was Mexico and other was Philippines. That means I can say two independent international institutes. One is CIMMYT and another one is IRRI. In 1968, Indian agronomist S.K.D. Datta published his findings of IR8 rice, which yielded about 5 tons per hectare with no fertilizer and almost 10 tons per hectare under optimum condition, which was 10 times the yield of our indigenous rice varieties. IR8 was a success throughout Asia and dubbed the miracle rice. It was also developed into semi-dwarf IR36. With the advances in molecular genetics, the mutant genes responsible for Arabidopsis thaliana gene, that is GA20 oxidase, GA1, GA1-3, wheat reduced height genes like RHD1 and RHD2, then rice semi-dwarf gene SD1 were cloned. These were identified as gibberellin biosynthesis genes or cellular signaling component genes. Stem growth in the mutant background is significantly reduced, leading to the dwarf phenotype. Photosynthetic investment in the stem is reduced dramatically as the shorter plants are inherently more stable mechanically. Assimilates become redirected to grain production, amplifying in particular the effect of chemical fertilizers on commercial yield. The technologies involved in green revolution. New varieties of wheat and other grains were instrumental to green revolution, whereas maize was excluded. The maize cultivation popularized only in the post era of green revolution, and it has got some importance only when the Indian farmers accepted it to feed the poultry sector as well as the livestock. The Green Revolution spread technologies that had already existed before but had not been widely used outside industrialized nations. These technologies included modern irrigation projects, pesticides, synthetic nitrogen fertilizers, and improved crop varieties developed through the conventional science based methods available at the time. Grain mountains and hungry millions. This is the growing paradox of post green revolution. The initiative in the form of Grain for Green movement, such program could accept or accord priority to restoration of hydrological and biodiversity hotspots, particularly in mountain ecosystem, coastal agro aquafarms like planting of Solicornia mangroves, Casuarina palms, etc., along with coastal agriculture and aquaculture, water harvesting, watershed development, wasteland reclamation, and anti-diversification measures, recycling of solid and liquid wastes and composting and agroforestry, and other sustainable land use systems in the field of resource poor farmers. Here why I am discussing about the grain mountains and hungry millions, because the approaches whatever I have discussed here are just to feed the hungry world. Coming to the impacts or positive impacts of green revolution on our Indian system, uh, I just want to tell you the green revolution adoption in India was very fast. For Mexico, green revolution happened in Mexico, Pakistan as well as in India, and for Mexico to adopt this new technologies, and to implement the green revolution they took around 18 years, whereas in India within 3 to 5 years it took widespread and became very popular and even fed the hungry Indian people. The positive impact is drastic changes in socio-economic political system. The imports from other countries and the economic burden for importing drastically reduced. Improved exports and country entered into global market. Increased production, improved transportation and irrigation, new varieties and hybrids of different agronomic and adverse climatic conditions. The negative impact is decreased food quality, desi variety erosion, land degradation, health hazards, decreased forest area, passively contributed for the global warming, and the diversity was drastically reduced, or we can say the genetic erosion was mainly found in the Indian crop system. The major aim after the or we can say after the green revolution, were genetically modified seeds to double the per acreage production, that is technology, then private sector to develop and market the usage of GM foods, that is efficient marketing of the ideas. The third kind is linking of rivers as much as economically possible to bring surplus water of one area to others, that is linking of rivers. Coming to the second green revolution, the term was coined by the Indian veteran Prime Minister Mr. Dr. Manmohan Singh. Here, poor investment in irrigation infrastructure, production has remained stagnant and yield in wheat, flat growth in production and yield of rice, fluctuating production and yield of oilseeds, no improvement in production and yield of pulses, 14% dip in per capita availability of food grains over the last 20 to 25 years. Agriculture sector faces lack of adequate funding, continuously rising population over the years, inadequate remuneration of the farmers, lower grain at government godowns, food subsidy in mounting, MSP, that is minimum support price, need to be increased. The recent facts are food security, food safety, food quality, and their linkages with poverty in the millennium. To achieve 4% annual growth in the next 5 years is the major challenge for Indian farmers. In 2010, the high time to bring reforms in the primary sectors to enforce efficient farm linkages, but it is still going till today. Technology development process of modern time required by the farming community is cooperation at local levels, regional levels, national um, level, at lastly at global level. It is very important to bring all national international opinions to achieve something. To keep the food security basket full, farmers require long-term agriculture policy for accurate planning by adopting modern infrastructure with the support of National Food Security Mission, Horticulture Mission, and low-interest financial support for long-term. Introduction of farmers pension scheme with the supply of every ton of food grains to state and central kitty by contributing 25% from his own share and 75% by state and central government for the minimum period of 15 years. Introduction of water and carbon credits to support the farmers in maintaining soil health and ecological imbalance by introducing short duration forestry like poplars and eucalyptus. Massive support system in procurement of food grains with a high quality production cereals like quality protein maize and protein wheat with the price on the basis of separate parameters. In the second green revolution, biofortification is a major area where we can concentrate. There are many biofortified crops which are already released, that may be in cereals, pulses, oilseeds, or some other crops. The government has declared recently drumstick and tamarind as naturally biofortified crops. The scope of problems which we are facing today, like high temperature damage, new pest and disease, altered drought pattern, effect of pollutants, constraints of solution strategies like varying rainfall distribution, frequency of occurrence and distribution of events of weather may change, dwindling agricultural low land and water, demand of land for livestock feed and bioenergy, economic and environmental costs of inputs. This picture shows the global impacts on productivity because of the increasing temperature or the climatic changes. The productivity of the land is declining continuously, though it may be slow, but the impact is high over the years on the ever growing population. The future challenges in agriculture are increase in world population, global warming, that is fluctuations in climate, limited availability of land, waste, water shortage and waste management. There is a need to prevent environmental degradation of land and water. need to increase food production under changing climate. Success in production or producing more food is only done through smart crops. This can be achieved through resilient crop varieties, input use efficiency, and adaptation. That is, the principle is more with less. The problems of desired smart crop varieties are they should be genetically diverse, suited to a range of agro ecosystem and farming, resilient to climate change, high yield, extreme weather conditions, efficient input use, conservation agriculture with zero tillage, multipurpose crop varieties. The areas for reorienting crop improvement are first one is broadening genetic base. In this, we should ask certain questions to ourselves like what should be the objective of breeding, how the crop is bred, where breeding should be suited in research and development, who are the main stakeholders in crop improvement, by whom, profit of breeder, then strengthening the national agriculture research and extension system, finally the organic plant breeding. Only with the commercial crop production we are not going to achieve anything like to be competent in the international market and to be sustainable in the countryside. We have to manage all the kind of systems. That means we have to efficiently utilize the different kinds of farming systems, or we have to go for the combination of different farming systems. Unlocking the inherent potential of PGRFM. In this, easy gene discovery, improved technology of breeding and genetics, then selection and hybridization that took 10,000 years, narrow genetic base. In Russia, 96% of winter wheat that is covered with only single or two kind of varieties that greatly created narrow genetic base. For this, the solution is widening source of variation and induced mutations. There is a need of understanding molecular basis of key traits, expanding the phenotyping and genotyping, molecular understanding of recombination, enhance the rate of introgression, and new breeding strategies for introgression. The priority activities of the second green revolution are in situ conservation and management. In this, we may go for surveying, supporting on-farm management and assisting farmers in disaster situations. Ex situ conservation strategies like collection, sustaining, regeneration and multiplying. Sustainable use with expanding characterization, evaluation, supporting plant breeding. Then building sustainable institutional and human capacities like national programs, information system, human capacity and public awareness. Coming to reinvigorated crop breeding for a changing world. In this, it is a business as usual mindset. As we can say, the modern agriculture or modern breeding strategy follows this principle. In the 20th century, we were having confusion with adaptation, quality, yield, and different other aspects we can say. But in the 21st century, we are clear with all those things. Here there is participatory breeding, demand-driven breeding, and multidisciplinary approach, which is enhancing the agriculture production as well as the total adaptation in the given area or with the same area what was available in the 20th century. Participatory plant breeding, factoring in the respective or perspective of growers and stakeholders like vendors, extensionists, industry and rural cooperatives in crop improvement endeavor. Involvement of farmers and end users in three stages. One is design stage. In this, setting broader goals. In testing stage, that is variability creation, and diffusion stage, that is narrowing down to few. The novel crop breeding techniques are objective of breeding for stress environment is to accumulate genes for stress tolerance naturally present in gene bank where water deficit, excess heat and salinity conditions exist. The constraints in breeding for abiotic stress are complex nature of the traits like time, duration, intensity and frequency. The quantification is quite difficult here. Then transfer of undesirable genes and reproductive barriers, that is nothing but biotechnological interventions. The key biotechnological strategies for improving crop for changing climate are: the first and foremost is molecular breeding approach, which became very popular in the 1970s until today. After even the invention of genetical modifications as well as the editing technologies, molecular breeding holds its own importance. The genome sequence of many crops which are available now, the next generation sequence, which made many of the genome sequencing cheap and quick. Genome sequence plus genetics, that led to identification of genes like sorghum, which is well adapted to stress, and identification of QTLs that can be transferable across the family, even across genera. Coming to the genetic engineering approach. Breakthroughs here: identification of signaling pathways, regulatory genes and networks of complex states, expression of several genes at required place and time, transcription factors like in rice, MB cofactor sulfarase, that is for ABA biosynthesis, AP37 to increase the grain yield, CBF3 or DREB1A, that is for drought tolerance. RNA chaperones, that is mediate transcription and translation, increase in yield under multiple stress, example bacterial cold shock protein, that is CSPB. Aquapores, these are present in tobacco to prevent shoot and root hydraulic failure, that was transferred to tomato to increase water use efficiency. Coming to the overall approaches in molecular breeding and genetic engineering. In molecular breeding, development of genomic sources or resources, development of biparental mapping populations for drought stress or any kind of biotic or abiotic stress breeding, the use of association mapping approaches, then breeding by design, validation of QTL markers in the breeding program, breeding as you go, then use of appropriate marker assisted backcross approaches, marker assisted recurrent selection and genome wide association studies. The genetic engineering approach, in this identification of genes encoding signaling proteins, transcription factors and effector proteins, identification of gene regulating stomata opening, genetic transformation and development of allied crop genotypes, assessment of promising transgenic lines and deregulation of transgenic lines to enable the release of a superior lines. Here the policy matters. Genetic engineering approach is very popular in the 21st century with the invent of golden rice and flour tomato, but because of it failed to prove the long-term safety issues, it is not much popular nowadays. So the editing techniques came recently and these two are considered as same, and there is a need of long term regulations as well as long term experiments to prove that genetic engineering is safe. If it is so, or if it will be done, many of the pesticides will go back and we may have an healthy environment in order to sustain our agriculture production. Here there are different methods of breeding. First one is conventional varieties, maintenance under conventional variety, then multiplication and conventional seed and vegetative propagation material. From there we may expect organic farming varieties and all. Then genetic engineering and which are prohibited, that we can't speak now. The third one is integrated approaches. Beyond molecular breeding and engineering, the best example is maize research in Africa. WEMA, that is water efficient maize in Africa, DTMA, that is drought tolerant maize in Africa, IMAS, that is improvement of maize for African soils. The CIMMYT bred high yielding maize cultivars expertise in conventional breeding with the utility of germplasm across the world, advanced breeding tools, and drought tolerance genes. Monsanto, Pioneer, and other private companies are also having big hand in this particular achievement. Here, with this advent, the crop improvement was 20 to 50% more than the integrated approach mainly work for changing climate, that is climate resilient agriculture, with the help of all the things like genetic engineering, molecular breeding, integrated breeding, sequencing, and all those approaches. Areas to touch upon the increased crop productivity are producing greenhouse gas emission, taking heat, drying out, carbonating the atmosphere, improving photosynthetic efficiency. These should be controlled. Reducing greenhouse gas emission. In this, decreasing the level of greenhouse gases like carbon dioxide and N2O along with methane. Reducing these from grazing animals, soil and plants, boosting the capture of carbon dioxide. Nitrous oxide emission. Here, nitrogen applied to soil in the form of fertilizer, manure and crop residues reduce nitrate leaching and volatilization of ammonia. The key area of genetic approaches to increase nutrient use efficiency to allow lower fertilization approach. Oats require less nitrogen among forages, increasing efficiency of digestive process like plant breeding with animal science. The high sugar grasses reduce nitrogen excretion. 9% improvement in water soluble carbohydrates in ryegrass that gave 39 to 26% decrease in nitrogen excretion. Exploitation of legumes for nitrogen use efficiency is quite better option. To shorten the methane emission, 21 to 25% methane is emitted from agriculture. The predominant source is ruminant fermentation. Quality diet of WSC reduces production of methane, for this increasing the oil content of conventional oats by 25% reduction in methane. Coming to taking out of heat. Decreasing yield due to high temperature. This we know, this is even experienced by us in the recent years, that lead to longer growing seasons in the low altitudes. Shift in climate cannot be considered to shifting cropping systems. The effect of this are increase in the rate of carboxylation by rubisco, increasing the solubility of oxygen and loss of carbon dioxide through photorespiration. Mitochondria respiration decrease, that is C3 and C4 decreased photosynthesis. Chloroplast protein, that is rubisco activase, also decrease, then increase in evapotranspiration. This effect can be mitigated by the genetic improvement technologies like gene shuffling to improve thermal stability of rubisco activase, lowering photorespiratory flux, and engineering foreign algal rubisco. To avoid the drying out, the drought is an important factor to limit the production. Almost 30% decrease in water resources by 2050. Improving yield under drought is difficult with low heritability. Many of the genes tolerant to drought found to be saline tolerant. This is the best information. The genetic improvement for drying out is, first one is ability to survive under water stress. that is with higher osmotic potential, closing of stomata, decreased cell dehydration. Stay green trait is to maintain the crop canopy green for a longer period, and that makes high photosynthate accumulates even in the high temperature condition. This is popularly called as sit out strategy. The heat found to be increase the yield under drought and salinity stress. Underground solutions in surviving rice plants. This is a study. A gene that enables rice plants to produce around 20% more grains, increase uptake of phosphorus, important but limited plant nutrient, PSTOL, that is Pestol 1, which stands for phosphorus starvation tolerance, helps rice grow a longer, better root system and thereby access more phosphorus. Improving water use efficiency. This is easy to select for less variable, explained by using on the basis of diffusion. C4 have less intracellular carbon dioxide with higher diffusion. C3 need to be engineered to photorespiratory bypass. This is already going in IRRI and many other international institutes to convert many of the C3 crops to C4 to increase its photosynthetic efficiency as well as to make it photoinsensitive. For this, many QTLs have been identified. The Erecta gene regulates plant transpiration efficiency in Arabidopsis. This experiment was done by Josette Messel in 2005. There is an ample scope for crop improvement with the conversion of C3 plants to C4. Achieving the maximum conversion efficiency by engineering Rubisco, RNAi or antisense technology, decreasing photorespiratory loss and plant architecture modifications. The winning partnerships in all these approaches are partnership beyond traditional national agriculture research and extension system is very essential in order to achieve this sustainable goal. The private sector is novel biotechnological tool source. Public sectors are ensured funding to work on untouched crops. Relevant private, public, CGIAR and regional research and rural networks. Then safeguarding the PVP laws and patents. Coming to national capacities for crop improvement. Survey of 81 countries through GIPB scope for funding, staffing, activities per capita. There is tenfold decrease in funding plant breeding activities between 1981 to 2001, but during these years recently it has again increased by 5 to 7%. A dying breed, that is current capacity for plant breeding, is inadequate. Training of the breeders, like to enhance the public-private partnership, training in traditional and other biological objects or subjects, public sector in which most graduates are trained, and in private sector provision for fellowships. The CGIAR centers are helpful in training in developing countries. Conclusion. In the whole video I explained the first and second green revolution, or else I can say the green revolution and the evergreen revolution. The main difference is only one. In the green revolution we gave more importance to the quantity of food production, because during that time the only aim was to feed the hungry people. But after that, once we achieved the self-sufficiency in food production, our main aim was to increase the quality as well as to tackle with the changing climatic situations in these years. For that, we expect that the second green revolution may help us in achieving the sustainability in the field production in the crop production, especially with the help of integrated approaches in breeding, agronomic or the crop protection measures. Thanks one and all.